it's my great pleasure to moderate this session on biologic and sociologic issues. My first speaker is Dr. Dean Fullman, who will be talking about the durability of responses to COVID-19 vaccines in adults and children. Since 2002, Dr. Fullman has been the Assistant Director for Biostatistics at NIAID, and his research interests include the development of novel statistical methodologies based on applied problems. It's my great pleasure to invite him to be the first speaker in this session. Thank you, Dr. Follmann.
Yeah, thank you, Catherine. I'm delighted to be here speaking today. There's been some great talks so far, and so I hope I can continue that tradition. I'm head of biostatistics at NIAID, and early on in the pandemic, NIAID and other people in HHS were marshaled to do many things, including be part of Operation Warp Speed to develop COVID-based vaccines. And so that's where I was. I was part of the Moderna trial, and I still continue to serve in that role, though we don't call it Warp Speed anymore. I think it's called H-Core. And so I'm going to give you that sort of perspective on the analysis of durability to vaccination. So I wanted to start off by just emphasizing that durability of immunity is complicated. There's a lot of different aspects to it. And when you read about it in the press or even in scientific magazines, it's maybe not so clear in terms of durability, in terms of what aspect you're talking about. So there are obviously players of the immune system, antibody, which we do measure generally, B cells and T cells, which we generally don't. And then there's the more clinically relevant, the clinical endpoints, which range in severity from infection to disease to severe disease or death. And durability for all of these can be different. Also, SARS-CoV-2 is evolving, as we well know, and so it's important also to couch durability in terms of what variant that it's being assessed against, ancestral, Delta, Omicron, or the new version variants that are coming out. And as we get increasingly sort of antigenically experienced through vaccination and natural infection, that's another player in terms of durability. And then also, finally, in terms of what population we're focused on, I could add sort of immunocompromised people as well. So it's a very complex arrangement here. I'll be focusing mostly on disease, which I think is what is most important. This is what the vaccines are meant to prevent. And so these are most meaningful, these clinical endpoints. The problem with those is that they can be slower to evaluate because it takes time to accrue cases and on and on. Antibody is relatively faster to measure, and so it's used quite commonly, but it's a surrogate for disease, and so it's not really disease necessarily. It's a proxy for what we're really interested in. So let's go back to November of 2020 when the first results were announced for the mRNA vaccines. Within a week, Pfizer and Moderna both showed or demonstrated that there were highly efficacious, 95% efficacy. This was like more than expected. On the right-hand side, there's a picture that I took on that day when I found out on 9 November that the Pfizer results came out. And so they're sort of evoking that there's hope coming through the fog on the Potomac River there. So the Moderna trial continued to follow up people for a period of time. And on this slide, I've got the New England Journal report of vaccine efficacy during the blinded phase of the Moderna trial, which ended in March of 2021. On the top panel, you see the accrual of cases. This is disease or COVID-19 in the placebo arm compared to the mRNA arm. And over this extended period of follow-up, there's 93% efficacy. In the lower panel, we have severe COVID-19 cases. And here the efficacy is even larger at 98%. And this is sort of a theme of the COVID vaccines. They have their bigger effect for more severe disease, maybe lesser effect against infection or asymptomatic infection. So these are sort of the top line results. On the right-hand side, we have a breakdown of the vaccine efficacy for different time intervals. And what I'd like to focus on here is the bottom, where it's broken up into different buckets of time. Overall, after 14 days following the second injection, the vaccine efficacy was 93%. And then it hovers around that for 14 days to less than 2 months, 2 months to 4 months, and 92% at greater than 4 months after the second injection. So things were promising. One of the things that was central to the warp speed trials was to look at durability of vaccination. We didn't know how long the vaccines would work. We knew from seasonal coronaviruses, immunity doesn't last that long, maybe up to 6 months or so. And for MERS and SARS-1, antibodies are detected longer after natural infection. But understanding durability was a key tenet or a key principle for the warp speed trials. So the trials were designed to continue the placebo arm for up to 2 years following enrollment. But this was quickly sort of realized to be ethically untenable. These altruistic volunteers had agreed to be in a trial, and through their contribution, we identified that the vaccine had worked. And so it made sense that they should sort of have access to the vaccine just as everyone else did. And so our hope for analyzing, or the idea that we could look at durability over 2 years with a placebo arm was out the window. And it seemed like that would really make it difficult to look at durability of vaccination. We don't have a placebo arm anymore. But there's actually a maneuver that we worked on that allows one to continue to assess durability of vaccination. And I'd like to sort of run it through in this schematic here, where we randomize people to vaccine or placebo, which is straightforward. In the vaccine arm, I have this thick blue triangle that's sort of getting smaller and lighter over time to denote the possibility of waning immunity. And in the placebo arm, they're getting placebo, so that's just an orange flat bar. After some point in time, early efficacy could be established, and it was for all the vaccines. And then for the Moderna trial and other trials, there was a period of time during which the data was evaluated. And after that, the FDA gave a green light in terms of an EUA, which opened up the vaccination for the other people for the placebo arm. And so you can continue the experiment, actually, and you can do it in different ways, an open label way. What I've suggested here is a blinded crossover where the original placebo arm gets the vaccine, but they don't know it, and the original vaccine arm gets a placebo, but they don't know that either. This was used actually in the Novavax trial. Some of the other trials just had an open label deferred vaccination. But importantly, if you look at the right-hand side, we still have a valid randomized comparison between two different interventions. Early on, this is a comparison of vaccination versus placebo. And now it's a randomized contrast of recently vaccinated and those who were vaccinated a while ago. And so if there's a difference in cases here, it has to be because the vaccine is losing durability. So we can tell if the vaccine is losing durability, but it's also nice to know whether, in fact, to also try and estimate what the actual efficacy is. And so here I'm going to show this schematically, where in period 1, we have a trial where we have 40 people in the vaccine arm, 40 people in the placebo arm. And at period 1, there's about 80% reduction in the case rate on vaccine compared to placebo. So we do that, we understand that we need to vaccinate the placebo group. And so we do that in the context of a crossover. So anyway, here's in period 2, we have the freshly vaccinated arm, which are dark blue. We have those who were vaccinated a while ago, the original arm. And so there's more than twice as many cases in the original arm who's been vaccinated a while ago compared to the deferred arm. So we know that the vaccine effect is waning, but we'd like to know whether it continues to have some benefit or not. And so what you can do is reason as follows. In period 1, there is 80% efficacy. And so to achieve 80% efficacy for the newly vaccinated in period 2, if we had a placebo arm, it would have about 5 times as many cases. This is what gives us an 80% vaccine efficacy for the freshly vaccinated. And with that, we can infer the durability of the vaccine. So I had a slide which showed that in the Moderna trial, there was durability of the initial vaccination up to about 8 months. But after that, the Delta wave hit, and we could no longer use that maneuver I showed on the previous slide to try and estimate durability. And so we're led to using observational studies. This is a result in the New England Journal by Dan Yulin and colleagues who sort of constructed a study where he looked at the cases and vaccination records within North Carolina. And here we see for the 3 different vaccines that are common in the United States, the Moderna vaccine, mRNA 1273, the Pfizer vaccine, BNT162b2, and the J&J adenovirus vector vaccine, the durability over time. And we see some waning of clinical efficacy here during the ancestral and Delta strain period in terms of clinical endpoints. One thing I want to point out here, though, is this study accrued cases from December of 2020 until September of 2021, and then it takes a while to get the data, to clean it, et cetera, and then it came out in 2022. And so the Delta, I mean, that's 2 variants ago, and this is sort of a recurrent problem with some of the observational study or of clinical endpoints. They're always lagging. Clinical endpoints is sort of a lagging indicator, and what's published is usually for a variant that had happened a little while ago. Something that's a little more quick that we can evaluate more quickly is neutralizing antibodies. And so as we know, vaccines induce the immune system to make antibodies to parts of SARS-CoV-2 and other things as well, but it's thought that neutralizing antibodies are the main mechanism of protection. And here's a little schematic of this where we have these blue Y-shaped proteins that recognize the SARS-CoV-2 spike. They attach to that and glom onto it in a way that prevents the virion from infecting a host cell, and thus infection is neutralized. And we can measure this neutralization ability within a cell. And there's an assay that David Montefiore at Duke worked out for HIV, which was adapted for SARS-CoV-2. And I illustrated here a pseudovirus is constructed, which looks like, which has spikes from a circulating strain of interest, maybe the ancestral strain, maybe now it's the Omicron strain. It's put in a well with infectable cells. And the pseudovirus has also been manipulated to have a firefly gene in it. And so if it is able to infect a cell, the lights turn on in that well. And so the intensity of light is a measure of how much pseudovirus is infecting infectable cells. If you put antibody in the well, this will abrogate or neutralize that infection process, and the lights will be off. And one can do this at different dilutions and come up with a measure, the ID50. It's the amount of virus that achieves 50% of the maximum luminescence. So this is the assay that's commonly used within the warp speed trials and what they've warped into the assay that was developed at David Montefiore's lab and continues to be used. And one important analysis that came out of this was to correlate neutralizing antibody with protection. So this was a paper that came out earlier this year, led by Peter Gilbert and a host of others, where we're able to measure pseudovirus neutralization, the ID50 point. This green histogram here is the distribution of neutralization antibodies that were achieved by vaccinees. Here's a nice strong correlation showing that more antibody results in higher protection. And at 100 ID50 units, we have about 90% protection. And so this evidence from the COVE trial supported the use of antibody as a proxy for clinical endpoints. So variants are happening, and here's an earlier paper showing how 2 doses of the Moderna vaccine, the antibodies wane over time, and we can do the pseudovirus neutralization assay for all sorts of different variants. And we see that for beta, for example, here, this this red thing, it starts out low and goes lower. I've graphed here also that 100 unit line, which is associated in the ancestral era using an ancestral pseudovirus with 90% protection. And this is sort of a benchmark or a reference for what we think might be needed for protection. So antibodies decay over time, and so boosting is a way to increase antibodies. And here's an experiment that was done where we have the Moderna vaccine. We achieved nice high titers of around 1,000 1 month after the second dose for the ancestral strain, and then they declined precipitously over a period of 7 months. The boosting reinvigorates them to near the original value, and then they have a more leisurely decline after the boost. Omicron, there's not as much of an antibody response to Omicron for the Moderna vaccine. They decline over time, but you get a big benefit from the boost, which then declines over time as well. Once again, here I have this red line. This corresponds to that ID50 of 100 units. So SARS has evolved and will continue to evolve, and so we're going to keep playing catch up with it, and we want to try and understand the best way to think about boosting and different variant vaccines. And so currently there's an NIH study called the COVAIL study, which is trying to investigate how the second booster dose might fare and what are the immune responses to the different variants with different flavors and different combinations for these different vaccine candidates. So this first arm would denote 3 doses of the original Moderna vaccine, followed by a boost by prototype. Here's a situation where the second boost is a combination of beta and Omicron, and so on. And we're trying to cover the waterfront here in terms of vaccines that are either available in the US now or likely to be licensed. The Sanofi is a protein-based vaccine, and so we're trying to understand this space as broadly as we can. So in summary, the vaccine protection waits for the clinical endpoints, which we've seen and continues to be the case. Protective efficacy is longer for severe disease and less for infection. And a problem with this, even though it's the most relevant endpoint, it's what we care about, is that we learn about it generally months after a new variant has emerged, or it's always sort of playing catch up. So we turn to neutralizing antibodies to help get more real-time decisions. I've shown how neutralization antibodies correlate with protection against disease. We can quickly assess durability of the antibodies by measuring it over time and also evaluate them using variant vaccines, as I illustrated in the COVAIL trial, which is ongoing. The use of antibody, though, as a proxy is really predicated on this assumption. It's more than an assumption, but it was demonstrated in the ancestral era that 100 IU per ml is sort of a bar that achieves 90% protection. And current work is trying to analyze data for this for the Omicron and Delta era. And then the future will be evaluating variant vaccines using variant neutralization assays, and this will help plan for future booster vaccination timing and strain selection. So with that, I'll stop, and thank you for your attention.